Today we will be talking about ELISA, a type of immunoassay. Immunoassays are a group of laboratory techniques that use the antibody-antigen reaction to detect and measure target molecules within a biological sample. An antigen is any molecule that produces an immune response in the body. Oftentimes these are foreign substances, but not always. An immune response refers to the generation of antibodies. Antibodies bind to antigens via the antigen binding site to form an antibody-antigen complex, as shown here. This allows the body to detect and eliminate the antigen. In natural settings, the body will produce a number of different antibodies that all bind to the antigen with varying specificity. However, antibodies developed in the lab, known as monoclonal antibodies, are highly specific and sensitive. Researchers take advantage of this interaction to rapidly detect molecules of interest in a laboratory setting. Today we will focus on one specific immunoassay technique called ELISA. ELISA stands for Enzyme-Linked Immunosorbent Assay. It is a very common and powerful assay that can detect the presence and quantity of antibodies, proteins, and other molecules in your biological sample. There are a number of different variations of ELISA. Let's first take a look at direct ELISA. Your biological samples will be loaded into a multi-well plate, as shown here. In each well, the molecules in your sample are coated or stuck onto the surface of the well. This includes your target antigen, if it is present. In this diagram, the target antigen is a protein. You then incubate the well with an enzyme-labeled antibody that is specific to the target antigen. This is where the antibody-antigen reaction is important. Recall that lab-made antibodies are highly specific. It will bind only to the antigens it was designed for, forming an antibody-antigen complex. The solution is then washed to remove excess antibody. If the target antigen is present, bound antibodies will remain stuck to the well. A substrate that reacts to the enzyme-labeled antibody is then introduced. The enzymatic reaction causes a color change. Let's take a closer look at how this enzyme-linked antibody works. The antibodies used in ELISA are labeled with or is attached to a specific enzyme that is able to react with a corresponding substrate. When the substrate is added to the solution, the enzyme modifies the substrate to cause a color change. The degree of color change depends on how much enzyme is present, which in turn is dependent on how much antigen is present. The degree of color change is converted into a numeric value to quantify the amount of antigen present in the sample. As seen in this example, the more yellow the wells appear, the higher the concentration of the target antigen present in each sample. Some common substrates used for ELISA are horseradish peroxidase and alkaline phosphatase. There are many variations of ELISA, and the exact use of antibodies and antigens can be modified to change the specificity of the test. One such example is the indirect ELISA. Here, there are two types of antibodies used, the primary and secondary antibody. As with the direct ELISA, the sample molecules are coated to the well and an antibody is introduced. However, in indirect ELISA, this primary antibody is unlabeled, meaning there's no enzyme attached. The primary antibody binds to the antigen if it is present in the sample. A secondary antibody, which is labeled, is then introduced. The secondary antibody targets the primary antibody rather than the antigen, so there is one additional step. The solution is washed and the substrate is then added, which reacts with the enzyme on the secondary antibody to form a color change. Indirect ELISA eliminates the need to label all your primary antibodies. This increases versatility, as secondary antibodies can detect multiple primary antibodies at the same time if designed to do so. This also increases sensitivity, as the primary antibody is better at detecting the antigen without the burden of the linked enzyme. Another variation of ELISA is sandwich ELISA. Here, the antibody to the target antigen is coated onto the well, not the antigen itself. This is then incubated with the target antigen from your biological sample. Next, you add an enzyme-labeled antibody that also detects the target antigen. As you can see, the antigen is sandwiched between the two antibodies. In larger target antigens, such as proteins, the two different antibodies often target two separate areas of the antigen. Substrate is then added, which reacts with the enzyme to form a color change, as we've seen before. Sandwich ELISA is more specific than other forms of ELISA, as it combines two different types of antibodies that both bind to the target antigen. It is used when high accuracy is required. Thank you so much for listening. I hope you now have a clear understanding of how the different variations of ELISA work.